techniques laboratory, engineering techniques in biotechnology laboratory, code 3708 here. So herein we will keep a target of doing at least 11 to 12 experiments. We will keep a target of 12 if time permits. So usually we have seen that around 10 or 11 or sometimes even 9 experiments are being conducted. But we will keep a target of 12 experiments and we will conduct it if the time permits. So in these experiments, first we will start with colorimetry, then slowly we will be getting into the spectroscopy because colorimetry it forms the basis of the spectrometric technique. When we talk about colorimetry, we talk about a specific three wavelengths, say red, green, and blue. But these color bands are also associated with one what you call this wavelength, right? For red, you have one wavelength, green, you have one wavelength, blue, you have one wavelength, right? Now, when we go into the spectroscopy, this wavelength scans it increases. Okay, so that's why we will start with colorimetry. We will see how different objects, say food products, leaf, whatever thing you can think of, you can analyze. You can also analyze your skin color and all these things using this device, and you see the changes. How uh, when you put different biological samples into the instrument, how the color changes. Okay. Now, in my last theory class, I have been discussing about this color as a qualitative and quantitative parameter. So here we will basically look into the quantitative parameter of the color and how we can use that quantitative parameter. If time permits, we can use some AI algorithms maybe in one of the future classes using this colorimetric device, and you can look into how you can integrate the artificial intelligence technique into the analytical techniques domain, right? So apart from this, we will be doing visible spectroscopy. We will be doing UV visible spectroscopy for the solutions, which most of you will be needing in your next year laboratory experiments when you go for the what you call this project, right? Then we'll be doing this, let's say DSC, then which is a thermal analysis. Before that we'll be doing FTIR analysis, which is Fourier transform infrared spectroscopy. So after that we'll be doing this colorimetry. Then क्या बोला था इससे पहले? Mechanical testing will be there. Then we will look into the moisture content analysis. Then different microscopic techniques. We will see how bright field microscopy is different from dark field microscopy, which is again different from polarizing microscopy. Okay, and what information they give you, right? Then we will look into the operation of the multiplate reader, how the multiplate reader it functions. Okay, then so along with that we will be looking into other things also. So these are some of the major experiments we will be discussing. Refractometry, did I tell you? We will be doing refractometry also, or different samples, and how refractometer it helps us in analyzing various food samples and other biological samples. Okay, so with now coming to the what do you call this hmm, evaluation, how you will be evaluated. So we will give you a format for report writing. So once you do this thing, today's experiment, you have to submit your record by next start of the class. Okay, as soon as you enter, you have to submit your record. So there are you can make the record in two ways. One is you can we have asked you to formulate 6-member group, at the max 6-member group. So what you can do is that that 6-member group, they write only one report, okay, and everyone owns that report. They have to sign, and they submit one printed copy. If anyone does not want to submit the print copy, they want to maintain their own record separately also, either in the this booklet form or in a single printed form. They are free to do that, because in the previous instances we have seen that in a group only 1% or 2% will work and others will not work. Okay, so why the person who is working, he or she will do all the hard work and everyone will take the share the laddu, right? So some students, they prefer, okay sir, I will, since I am doing all the thing, I will submit the record separately. So I am fine with both the things. Okay, so either you prepare in a group. If you don't want to work in the group and submit a separate report, you do it. Because it sometimes happens that you give some task to some other person, and in that group that person, he does not work, then it will affect all of your grades in the group, right? So whatever you decide on your own. Okay, then based on that records, we'll be having we'll be marking 10 marks every week. Then every week as you enter, just after entering, you will be shared with a Google form. There will be a 10 to 20 marks examination based on what you have done in the previous class. So for today's class, there will be a class test in the next week. Okay, the portal will be open for 10 minutes because it will be an MCQ based question. It will be open for 10 minutes. Whatever you answer, and you will immediately get your marks. Okay, so that will constitute the second thing. Then uh, TA will give you assignment. Okay, related to the whatever the experiment you are giving. Some theoretical assignments will be given. So that will have certain percentage of marks, right? Apart from this, if anyone wants to do, see, it's very uh, difficult for a student to get all the things and get a very decent year. If you can come up with any practical this what you call some small projects, it will carry extra marks. Okay, suppose you are not performing well in any of the tests and all these things, but if you come up with a decent project, I am ready to give you extra marks. Okay, but it has to be a functional one. You have the full semester with you. You think whatever you think of. I do not have any here. So that extra marks will be added if you come up with a functional analytical technique device or any project. Okay, mostly yeah, functional some device. So if you want any help, we are here to help you out. So, but you have to do it. So this would be the marking scheme. And do not copy ये आपका record books or the assignment. वो पता चल जाता है. So suppose you have done the experiment and you have given to your colleague, they will ditto copy it. वो full stop by full stop copy हो जाता है. So there are other techniques also by which we can understand who has copied. So, but we don't know who has copied from whom. So both will get zero. Okay. So be careful with that. In the previous, I am telling you the fact. In cases of the for your seniors, someone has given another guy, अरे तुम कर लेना लेकिन अपना language वगैरह सारा change कर लेना, right? But they have not changed anything. They have just ditto copied it. So don't do that. Don't trust anyone. Okay. So any questions for me? No. Chalo.
Okay, welcome everyone. Myself W Sav. So I'll be taking you this first experiments. It's about colorimetry. As sir already explained about this, how the colors is important for this biotechnology aspect. So I'm directly going into the theoretical part of it. If you go, uh, the color is nothing but the human's eye perception, how a human eye perceived the objects that are around us, right? So basically, if we go for the electromagnetic spectrum, the visible spectrum lies in between 400 to 700 nanometer. I think everybody knows. It's might be different in different literature. Like some books you will find 380 to 730 nanometer, and in some other books you will find this 400 to 700 nanometer, right? So to the main objective of the colorimetry is to it's uh, how to numerically represents the color of an object. Okay, so it has actually two ways how you will actually going to get the color information of an any object. Either you will go by this reflective spectrophotometry or colorimetry. We'll go how we'll differentiate what is the difference between these two. What is the reflective spectrometry and the colorimetry in the in upcoming slides. Okay, now coming to this reflective spectrophotometer, this is used to measure the reflectance or transmittance of a material across range of wavelengths. It it not bound to visible, but we can see in the UV range also. And if you guys have, I think you guys have studied in this physics and all, that UV ranges from your 190 nanometer to UV visible. If you are going for 390 nanometer to, it will come around 730 nanometer, right?
100.
Yeah, 100 nanometer. So
It
in
is the experimental UV 192 because below 190 also you have UV region, but it is difficult to make instruments which will be working under the laboratory equipment using and working within that in the UV region below 190. So for experimental purposes, it is usually 190 to 400, and from 400 to, in some books, as you said, it will be 730. In some books, you will find it is 780 nanometer. 400 to, at the max, 780 nanometer is the visible range. And working UV range is 190 to 400.
Yeah.
To 400 range, just below 400. 399, you can say.
Yeah. Now coming to the working principle of this reflective spectrophotometers, it's basically how the light is reflecting from an object. At like if you are seeing a green object, means I think if you will have studied in optics and all that, which light it's actually reflecting. Can anyone tell? If it is green, leaves suppose. All
Absorbing all and reflecting green.
exactly. So based on that reflection, we can predict the color of any object or any kind of sample that we are given, right? So actually what we'll get, like if we are getting some information about the color of a material, how this will be helpful for our study, that thing we will going to see in our upcoming slides, how this will be helpful for your like study for performing any kind of experiments and all. Now coming to the colorimeter, actual colorimeter, it's only for the visible range. It will quantify the amount of light that has been absorbed or transmitted by the sample at different wavelengths. And as I mentioned in the reflective spectrophotometers, it may lies from UV to visible, but for the colorimeter it has it's only for the visible range. Okay, this is one difference. Then coming to how this colorimeter works. Okay, it's the working principle of reflective spectrophotometer and colorimeter is little bit same. It has a source of light. Okay, you have sample. You have to like radiate the light or you have to give the turn on the light to get the reflection or reflected light back to the one detector will be there, right? So basically three components, let me clarify. One detector, then your sample, and your source. Okay, source will be radiate the light, it's fall on the sample and it will be reflected back to the detector. Okay, and that will be detected by the detector and you will get the wavelength information as well as your color information. If you are going for this transmission type, the arrangement of the detector and the source will be different. If you are going for the reflected type, the arrangement will be different. This thing we will get to know about more about this when we will study about this UV visible spectroscopy. Okay.
In simple words, if you want to see any reflective thing, suppose this is an opaque stuff, right? I want to see. So since some light is coming from my back, it is falling on the object and it is reflected to my eyes. So I am able to see this object, right? So this is what I am seeing. I am seeing basically the reflected light. So in reflective spectrometry, what you have, the detector and the source of the light, they are on the same side of the object. Whereas if you want to see through the glass and want to see what is on the other side of the glass, so that is transmission. So light is transmitted. On the other side, you have the light that is coming through the glass, and then I am able to see it. So that is that you can relate to the transmission spectroscopy. Okay. So in transmission spectroscopy, you have the light source and the detectors on the either side of the what you call sample, right? And in reflective spectrometry or colorimetry, it is on the same side of the sample.
These are the some of the difference between the colorimetry and yeah. As I mentioned that the difference between the spectrometry and colorimetry, here you can see what are the basic difference. Okay. Now coming to the different attributes that are there in the colors. Okay. First, here I have listed three main attributes, that is hue, saturation, and lightness. Okay. So what is hue actually? Hue is the hue nothing but the color that will describe how the color is what is the color of an object. Okay. What is saturation? Saturation is the purity, how pure is the color. Okay. Now if you go for this lightness or intensity, it will give you the information about the whether the sample is darker or lighter. Okay, these are the three things for the three attribute of the color itself, the what is the main color of the sample, then its saturation, which is nothing but the purity, then the lightness, or you may say the intensity. Okay. Now coming to the different color spaces, actually to describe an color, there are various color space. First one everybody knows RGB, right? Red, green, blue. The primary color that we use to get any color from that, right? So this RGB color space give you the information about it will uh, quantify the color of the object in three: red, green, and blue. Like that, we have other color spaces like CMY. cyan, magenta, and yellow. Then HSI, as I mentioned, hue, saturation, intensity, or you may say it is lightness. So there are some limitation in every color spaces. Like if you go for this RGB color space depends on its light, how the light is you are illuminating the sample. It will depends on the light intensity, everything it will depends on. If you reduce the intensity of the light, the RGB values will be changed. But to avoid that limitation, this HSV or we can say HSI color space came. Okay, if you see based on the human color perception, uh, here you have segregate three things like hue, it will give you the color information, saturation give you the purity of the color, and the I, it will give you the intensity of the color, which is we will not able to get it into RGB color space, right, where you will only get the color information like R, G, and T. Like that we have L star, S star, B star. We also call this color space as a CIE lab color parameters. Here we have divided this color into a 3D three-dimensional space. I'll show you how this color parameters will describe the color of an object in the coming slide. If you the L star, it's nothing but the whether the sample lightness, it will describe the lightness of the sample, right? So if you are getting towards 0 means it is darker, if you are getting towards 100 means the sample is it's lighter, you can say. Like that this A star and B star will give you the chromacity parameter of the sample. If you see here, this is a three-dimensional representation of this L star, S star, B star, this right side one, and this left side one is the HSI that I have discussed, right? Hue, saturation, and your intensity. If you see here, this uh, this is nothing but a circle, right? This perimeter, if you is it so, this is 0 degree, okay? If you are going anti-clockwise in 90 degree, you will find this yellow. Like that, all color will be covered in this circle. Okay, this is the chroma in the sense the uh, and saturation, yeah, exactly, saturation and intensity, it will give you the whether it is darker or lighter. Now coming to this uh, three-dimensional space, that is L star, S star, B star. Here you can see if you are getting a positive A star value, that represents there is a presence of red tint in your sample. Okay, if you are getting a negative A star, it will tell that in your sample there is green components that are present. Okay, like that if you are getting a positive B star value, yellow components are there. If you are getting a negative B star value, there are blue component. Or for the L star, it is nothing but the 0 is more darker and 100 is more lighter. Okay, and if you try to interpret this HSI into your this S star this CIE lab color space, you can do also, you can perform that also. This circle, if you draw a circle, that will represent your hue, then saturation and intensity. Now coming to this, some of the example that we have took here, like if you see this yellow rose, right? This is red rose. So we are perceiving that in our eye. Now what we will do, we will quantify these things in the instrument. Okay. If you see these two apples, can you see what is the, by seeing only, can you see the difference, right? Uh, there are some difference, right? But to quantify that, we in naked eye we can't do that quantification. How much color difference, or what are the physical changes that have been there in term of color, right? Because both are seeing red only. In naked eye we can't say these two apples are different. But when you perform the color analysis, if you see here, L star is giving 43.31, whereas for this apple, this L star is giving 47.34, right? Means although the two apples are seem to be same, we are getting different color information for both the apple, right? These are the this is the main reason why we need a color analysis device. Otherwise, in the naked eye also we can tell this is yellow, right? But when you are going for some biological experiments, the color might be little bit same, but that minute difference in color can explain a lot of things that we can't predict with our naked eye. Okay. Now, coming to most of the main application of colorimetry that we can perform, like it can be used in paint industry, fabric industry, like for getting fabrics and all. Uh, it's also used to determine the quality of food. Then, yeah, this is in term of biotechnology aspect. We can use this in microbial characterization and identification. Like if you go for this bacteria and fungi, their pigments are different. Okay. So we can easily means identify whether this is a fungi or bacteria through their colors. Okay. Now, in pharmaceutical industry also it is very widely used technique to get the color. Okay. Now, and yeah, for the development of the biosensor, I think everybody heard about biosensors. Any idea about biosensors? What can you explain? Biosensor? Yeah, like glucose measurement. Okay. If you see in that glucose measurement devices or sensor, you used to have this color analysis thing. When the glucose come in contact with that sensor, that color will be changed. Okay. So the to quantify that how much glucose is present, you need to measure that color of that sensor, right? So in that case also we can use this color analysis thing. But mostly in our lab, that the sample that we have took with means with you guys will take the reading of that sample. Actually, we have one commercially available device that we will show you in the upcoming classes, and we have developed one device in our lab for analyzing the color of the device. Okay, color of any sample. So we'll do it in the this device that has been developed in our lab. Okay, it will give you the information in 4 color space. You can write it down because you have to mention it in your record book. Okay, so it will give you the color information in 4 color spaces that I have discussed: RGB, HSI, CMY, and CIE lab color space. Repeating: RGB color space, CMY color space, HSI color space, and CIE lab color space. 4 color space: C I E capital LLAB. These things should be there in your record book. Okay, don't take it lightly, and don't take, yeah, RGB color space, HSI color space, CMY color space, then capital C, capital I, capital E, lab, LAB, color space. No space between C, I, E and lab, okay? That is one letter word. I will show you. Here you can see, okay? So the device that I am going to show you, it's based on, it's a reflective spectrophotometer, okay? Means as sir has mentioned, the detector and the illuminator are there in the same side, okay? We have the detector here, we have the light source here. Light source will illuminate the sample and the light will be reflected back to the detector and it will be detected through computer vision technique. We will get the color information of the sample. That's all, okay? So don't take the records that are from your senior and submit it, because I have took for the last 2 years or 3 years I am taking the classes for this lab. By seeing only I can tell, and we have the record book also, record book of your senior also. So don't take, because every years we are changing the things, like there are little bit modification in the sample analysis and all. So don't take it and submit it as it is, okay?